Moving on from our condensation polymers lesson, we'll look at another type of condensation polymer called a polypeptide. In this session, we're going to describe what polypeptides are. We're going to explain why polypeptides are a type of condensation polymer. And we're going to be able to draw polypeptide structures and identify the peptide link. Now, polypeptides are made up of many peptides, just like polymers are made up of many monomers. Many polypeptides joined together will create amino acids. Many amino acids joined together will make a protein. Proteins are used by our bodies to repair cells and for growth. Before working on polypeptides, we need to look at what a peptide looks like. So the simplest amino acid is glycine. So it's the easiest one to demonstrate the different groups on. So if you look at it, it looks like this: NCC. The last carbon has a double bond O, single bond OH, a bit like a carboxylic acid. This end has two H's on the nitrogen. The carbon has two H's on it as well. So that's the simplest amino acid, glycine. Two ends of it. The first end we need to look at here is an acidic side. Okay. On the other end we have our basic side. So it's a well balanced structure. Polypeptides are many peptides joined together. Now we call these condensation polymers because they form water as one of the products when they're joined together. So the easiest way we can join these two glycine molecules is by removing the water here. So you follow the structure, first put in the backbone, NCC. We know that this carbon will then be joined to this nitrogen here because we've lost the water in between. We then need to structure our peptide link. Now peptide links look like this. Because we've got a double bond O going up here, we'll put a double bond O going up here. Okay? Because we've got this hydrogen going up here, we could put the hydrogen above or below, it doesn't matter. I like putting it in opposite so it looks a lot better. You can put it either side, it doesn't matter. You could put the oxygen, double bond oxygen down here, you could put the single bond hydrogen up there, as long as you've got this structure in there somewhere. Okay? Then you just complete the rest of the structure. Okay. Remember that carbon has to have 4 lines coming out of it, otherwise it's incomplete. Finally, you will be asked to identify the peptide link. Now the peptide link is this structure here. So you've got carbon double bonded to an oxygen and a nitrogen single bonded to a hydrogen. Okay, so that's one peptide link. You may be asked to give a specific number of peptides in a polypeptide. So if they ask you in the exam to put 3 glycine molecules together, you would do it like this. Okay, so you'd put NCC, that's the first glycine, and then NCC for the second one, NCC for the third one. Now always finish off with the double bond O, single bond OH, and we know that this end is going to be single bond H two ends. Okay. In the center, you have to think what happens at the end of a glycine molecule. Okay. So we look at this one in the middle first. So we know that this nitrogen is going to have a H, and this carbon will have a double bond O. Okay. We do the same for this one here. So hydrogen, oxygen. We now just fill up the rest of the molecule. Okay, so we know that this carbon will look like this, and this one will look like that. Just remember that each carbon needs to have 4 lines coming out of it. Okay. Finally, they will ask you to identify the peptide link. You just put a box around it or a circle around it, doesn't really matter. There's one, and here's our second one. So there's 2 peptide links in this polypeptide. We can't forget about the water that's been released as well. So if we got 1 peptide link here and another peptide link here, we must have produced 2 lots of water. So 2 H2O has also been produced. Okay. If we had more peptide links, the number of peptide links will link to the number of water molecules that are produced at the end.